Hello viewers, we are going to discuss about transport across membrane, active and passive transport, facilitated transport. Before going into the detail, I would like to mention it is an initiative taken up by Zoological Society of Assam to support undergraduate students studying life science, zoology or botany. Now let us see the learning outcomes of this topic. After going through the presentation, a student will know briefly about the plasma membrane structure and composition, in detail about various mechanisms of transport of small molecules across the plasma membrane, different types of proteins involved in the transport process, structure and function of the proteins involved, and clinical issues if any. So now let us go into the topic. Different types of membranes are present depending upon permeability. Permeable membrane allows substances to pass through, for example the cell wall in plant. Impermeable membrane never allows any substances to pass through. Semi-permeable membrane allows certain substances to pass through depending upon size. Selectively permeable membrane allows the passage of some molecules or ions and inhibits the passage of others in which size is not a factor, for example plasma membrane. Both prokaryotic and eukaryotic cells are surrounded by plasma membrane, which defines the boundary of the cell and separates the internal content from the environment. The basic structure of plasma membrane of cell is the phospholipid bilayer, which is impermeable to most water soluble molecules. The passage of ions and most biological molecules across the plasma membrane is therefore mediated by proteins, which are responsible for selective traffic of molecules to and out of the cell. Other proteins of the plasma membrane controls the interaction between cell of multicellular organism and serve as sensors through which the cell receives signal from the environment. So the plasma membrane plays a dual role. It both isolates the cytoplasm and mediates interactions between the cell and its environment. According to the model proposed by Singer and Nicholson in 1972, there are two types of proteins available. One is called peripheral membrane protein and another one is integral membrane protein. Peripheral membrane protein, it may remain outside or inside, whereas integral membrane protein, they pass through the membrane. So it may be in the diagram there are two peripheral membrane proteins are shown. One is towards the periphery of the cell, one is inside the cell. On the other hand, if we look into the integral membrane protein, in the diagram again two integral proteins are shown. One is the glycoprotein and another one is the transport protein, which is forming a channel. The internal composition of the cell is maintained as the plasma membrane is selectively permeable structure. The plasma membrane forms a barrier that blocks the free exchange of molecules between the cytoplasm and the external environment of the cell. Most biological molecules are unable to diffuse through the protein-free phospholipid bilayer. Small hydrophobic molecules such as oxygen, carbon dioxide, nitrogen, benzene, or the small uncharged polar molecules such as water, glycerol and ethanol, they can pass easily through the membrane. Whereas large uncharged polar molecules such as amino acids, glucose and nucleotides and different ions, they cannot pass through the membrane. To carry the molecules which cannot pass through the membrane easily, there are some mechanisms by which the molecule can pass through the membrane. There is one mechanism in which the molecules can move from a higher concentration to lower concentration. In that case, it does not require any energy, and this is known as passive transport. Whereas in some cases, molecule has to move against a concentration gradient which requires energy, when the molecule has to move from lower to higher concentration, and in that case the transport is known as active transport. To accomplish active transport, there are some special type of carriers present at the plasma membrane. In the plasma membrane, there are two major classes of transport protein. One is carrier protein and another one is channel protein. Carrier proteins, they move the solute across the membrane by binding it on one side and transporting it to the other side. So it requires a conformational change. Whereas in channel protein, it is a small hydrophilic open pore which allows the solutes of appropriate size and charge to pass through. It uses diffusion to move across the membrane. They are also known as ion channels. Now, if we look into the image, we can see that how channel protein and carrier proteins are involved in the transport process. In passive transport, both the channel protein and carrier proteins are involved. Here, the molecules are transported from higher to lower concentration, so no energy has to be spent. Whereas in active transport, only the carrier protein takes part and the molecules are transferred from lower to higher concentration, so energy has to be spent in this process. Till now, what we can say, to carry small molecules across plasma membrane, transport can be divided into two types. One is passive transport and another one is active transport. Passive transport again can be further subdivided into simple diffusion and facilitated diffusion. In simple diffusion, molecule simply dissolves the phospholipid bilayer and diffuses across it. Whereas in facilitated diffusion, passage is mediated by a transport protein, which may be a channel or may be a carrier protein. Here, the rate of transport is much more than the diffusion. There is another process involved which is known as osmosis, in which the net movement of water is caused by the concentration difference of water. Now, let us discuss about the structure of membrane carrier protein. Here, we are going to discuss about the structure of glucose carrier. Glucose carrier consists of a protein chain that crosses the plasma membrane about 12 times, and it has two conformations, open and close. In human, this GLUT carrier has 14 isoforms, and they are having different substrate specificities for glucose and other hexoses. These 14 isoforms are present in different organs such as liver, kidney, etc. Now, if you look at the picture, what can be said, these carrier proteins, they are very much specific in nature. Suppose if we look at the plasma membrane, there in this diagram we could see 4 different types of carrier proteins. Each carries a different type of molecule. One molecule is there for carrying nucleotide, another one is for there to carry sugar, the third one is for amino acid, and the fourth one is for transporting sodium ions. In the same way, in lysosome there is another carrier, and in mitochondria, which is double layered, there are carriers, separate carriers for carrying pyruvate and ATP. So these carrier proteins, what they do, they cycle between conformations in which a solute binding site is accessible on one side of the membrane or the other side. There may be an intermediate conformation in which a bound substrate is inaccessible to either aqueous phase. Generally, a carrier transports one or a few solid molecules per conformational cycle, whereas a single channel opening event may allow flux of many thousands ions. Many ions can pass, or a large number of ions can pass through a channel, whereas through a carrier, comparatively a very few number of molecules can pass. That is the difference.
Glucose is transported from higher to lower concentration by GLUT1 carrier present in mammalian liver or in any other cell. When glucose level is abundant outside the liver cell, especially after a meal, glucose molecule bind to externally displayed binding sites, the state A. When protein switches conformation, it carries these molecules inward and releases them into the cytosol, where the glucose concentration is low. On the other hand, when blood sugar level are low, the hormone glucagon stimulates the liver cell to produce large amount of glucose by breakdown of glycogen. As a result, the glucose concentration is higher inside the cell than outside, and the glucose binds to the carrier protein. When the protein switches its conformation in opposite direction, the glucose is transported out of the cell. The flow of glucose thus can go either way. But these transport carriers are very specific. They bind only to D-glucose molecule.
The channel proteins, they create a hydrophilic opening in which small water soluble molecules can pass into or out of the cell. Among the channel proteins, the gap junction and porin, they make a very large opening. Whereas the ion channels are very specific with regards to pore size and charge on the molecule to be moved. They move mainly ions such as sodium, potassium, chloride and calcium. The rate of transport is again much higher than the carrier protein. Repeatedly I am telling, and they are basically concerned to transport inorganic ions, the ion channels. Ion channels, they are highly selective in nature. They allows some ions to pass and restrict the others. For a specific type of ion, there is a specific type of channel. Based upon their pore size and the charge on the inner wall of the channel, they filters which ions to go through the channel, and they are not always open. They have, when it's required, they remains open, and when transport is not required, then they remains in closed conformation. They have the ability to regulate the movement of ions so that control can maintain the ion concentrations within the cell. To do so, there are certain specific stimuli which triggers the change in shape and opening and closing of the channel. Now the types of channel. There are basically three types of channels available. The first one is the voltage gated channel, which operates in open and closed conformation when it is excited by a membrane potential. The second type, which is ligand gated channel, they are controlled by a ligand. When the ligand will bind, that time the channel will open, and when if the ligand is not bind with the protein, then it will remain closed. It may be, the ligand may be extracellular or it may be intracellular. And the Third one is the stress activated channel, which is controlled by mechanical force on the cell. Now the voltage gated channel. Here we can give the example of sodium channel. Sodium channel, it is mostly present throughout the length of the axon fiber. In axon fiber or in nerve cell, the sodium ion plays a crucial role in conduction of nerve impulse. When the nerve is not carrying an impulse or it is in resting state, outside the membrane it is positively charged and inside it is negatively charged. When the nerve is excited by an electrical stimulus, then the sodium channel, it from closed configuration it remains open. Since sodium concentration is high outside the cell, so huge number of sodium ion comes into the cell, disturbing its resting membrane potential. At the same time, through potassium channel, large number of potassium ion goes out. So that's how the sodium channel is open and in closed state due to the presence of an electrical stimulus or voltage. The other type of channel, which is a ligand-gated channel, that is also seen in nerve cell, especially in the synapse. Here we can give the example of sodium-potassium gated channel. Here also you can see in the second diagram that sodium-potassium channel there remains closed when there is no ligand. But when the nerve terminal is excited by the electrical stimulus, then what happens? The vesicles which carrying some neurotransmitter is forced to release its content to the synapse, and the neurotransmitters, they serve as a ligand and they binds to the postsynaptic cell in the sodium potassium channel, and when they binds, the channel remains open and huge number of sodium comes in and potassium goes out, and thus they disturbs the membrane potential, and the nerve impulse is transmitted from presynaptic cell to postsynaptic cell. Another type of channel is stress activated channel. A stress activated channel is controlled by a mechanical force applied to the channel. For example, the auditory hair cells present in the internal ear. A section through organ of Corti showing the auditory hair and nerve cells. The sound vibration vibrates the tectorial membrane, which is colored yellow, results opening of stress activated channels, due to which inflow of ions sets up an electrical signal which is transmitted from hair cells to auditory nerve and conveys the signal to brain. Now let us talk about the AQUAPORINS. They are specialized water channel proteins and they are highly tissue specific. Like GLUT carrier, they are also having different isoforms. They generally transport small uncharged molecules along with water, such as ammonia. They are impermeable to charged ions. They only allows water molecules. Then water molecules, they pass more rapidly through the aquaporin even than the phospholipid bilayer. They plays crucial role in plants as well as in animals. In plants, they plays role in transpiration regulation, whereas in animals it maintains water balance in brain and extrudes sweat from the skin. So it allows free passage of water without affecting the electrochemical gradient across the plasma membrane.
Carrier protein performs three types of transport. In uniport, the protein carries one type of solute across the membrane. For example, the glucose transport, which is mentioned just now. The second type of transport is symport, in which the protein binds two dissimilar solute and transport them together across a membrane. For example, glucose and sodium symport, which will be discussed in later part of this presentation. And the third one is antiport, which is also known as exchange diffusion, in which the carrier molecule carries one solute for another across a membrane. For example, chloride and bicarbonate antiporter, which is present in erythrocytes.
Now, active transport. There are total three methods by which the solute moves against electrochemical gradient. The first one is the ATP driven pump coupled to ATP hydrolysis. The second one is coupled transporter, in which one molecule goes down the concentration gradient and other goes up the concentration gradient. And third one is the light driven pump, where light is used as a source of energy, mostly in bacteria. So now let us see what happens in sodium potassium ATPase pump. It is also known as ATP driven primary or direct active transport. Here energy is required, and that required energy for the transport is derived from hydrolysis of ATP. In this pump, what it does, it keeps sodium ion 10 to 30 times lower than extracellular level, and potassium 10 to 30 times higher than the extracellular level inside the cell, due to which there is a sodium potassium gradient across the cell, which is very much essential for active transport of ion and maintenance of membrane potential. Here in the image, structure of sodium potassium ATPase pump is shown. It is an integral membrane protein having two subunits. One unit is 110 kilodalton alpha and another unit is 55 kilodalton beta subunit. Here, upon hydrolysis of ATP, the protein undergoes a conformational change. What happens? ATP breaks down into ADP and phosphate, and the phosphate binds to the protein, which leads to a conformational change. And if you see the membrane, outside the membrane sodium ion concentration is high, whereas inside it is low. On the other hand, potassium ion concentration outside it is less, whereas inside the cell potassium ion concentration is high. But both the ions, sodium and potassium, they are moving against the concentration gradient. When 3 sodium ion they goes out, at the same time 2 potassium ion comes in. So this is just an example. The animal cells, they maintain osmotic balance and membrane potential by sodium potassium pump. This pump works continuously and it cannot be halted. If halted by using some inhibitor, or in some cases due to absence of ATP, the cell may burst. How? Because if low concentration of solute outside the cell and inside it's high, then water molecules will come in by osmosis and cell will swell, and as a result the cell will burst. That's why this sodium potassium pump, they acts ceaselessly or continuously to maintain the ionic balance throughout the cell. Let us discuss about secondary active transport, which is also known as ion driven or indirect active transport. When sodium ions are transported out of the cell by primary active transport, a large concentration gradient of sodium ions across the cell membrane usually develops. High concentration outside the cell and very low concentration inside. This gradient represents a storehouse of energy because the excess sodium outside the cell membrane is always attempting to diffuse to the interior. Under appropriate conditions, this diffusion energy of sodium can pull other substances along with the sodium through the cell membrane. This phenomenon is called co-transport. Here we can give the example of glucose pump. Let us discuss about glucose pump. The cells lining the lumen of intestine have two distinct sides which have different lipid and protein compositions. The apical or brass border membrane facing the lumen, which is highly folded into microvilli to increase the surface area available for absorption of nutrients. The rest of the plasma membrane, the basolateral surface, is in contact with neighboring cells and the blood capillaries. Movement between adjacent epithelial cells is prevented by the formation of tight junctions around the cells near the apical domain. Thus, any nutrient molecule in the lumen of intestine have to pass through the cytosol of epithelial cell in order to enter the blood. The nutrient cannot pass to the neighboring epithelial cells. Energy for glucose movement against concentration gradient from lumen to epithelial cell comes from sodium ion movement down its concentration gradient. Glucose moves out of the cell to basolateral membrane by facilitated diffusion. Low concentration of sodium inside the epithelial cell is maintained by sodium potassium ATPase pump in the basolateral membrane. Glucose rehydration therapy. The movement of sodium ion and glucose from the lumen of the intestine across the epithelial cell to the blood sets up difference in osmotic pressure across the cell, due to which water flows through the cell across the apical and basolateral membranes by simple diffusion. Hence, the uptake of water requires both sodium and glucose to be present in intestinal lumen. The presence of water alone in the lumen of the intestine is much less effective. That is the basis of glucose rehydration therapy as a remedy of dehydration. Let us talk about some inherited ion channel diseases. These diseases are due to mutations in genes encoding channels. Disease due to mutation in chloride channel encoding protein is cystic fibrosis. Here, abnormally thick sticky mucus is secreted by the cells lining respiratory and gastrointestinal tracts. It is diagnosed by excessive salt in sweat. Here, pulmonary airways are obstructed by thick mucus leading to severe bacterial infection. In most of the cases, the patient dies within the age of 30. Another disease is the inherited tendency to kidney stones, which is caused by a different kind of chloride channel than the one involved in cystic fibrosis. In diseases involved mutation in sodium channel protein, we have to mention Liddle syndrome, in which inadequate sodium is transported out of the kidneys, leading to elevated osmotic pressure of blood and resulting hypertension. And there is a inherited tendency to certain types of muscle spasm. To summarize the transport process across plasma membrane, two types of molecules are involved. Here we have discussed only the transport process involving small molecules. There are two ways of transport. One is passive transport and another one is active transport. In passive transport, molecules flows from higher to lower concentration. Again, passive transport can be further subdivided into two, that is simple diffusion and facilitated diffusion. In simple diffusion, molecule can simply cross the plasma membrane, whereas in facilitated diffusion, some proteins are required which acts as a facilitator. This facilitated diffusion can further be subdivided into carrier mediated facilitated diffusion and channel mediated facilitated diffusion. If we talk about active transport, here energy has to be spent because the molecule to be transported has to pass from lower to higher concentration. Here the active transport can be further subdivided into two. One is primary active transport and another one is secondary active transport. For this topic of membrane transport, if you want to study further, then I recommend the following three books. Although there are many books are available in the market. So I would like to thank you for your kind attention, and if you have any query regarding the presentation, you can email me at the following email ID. Thank you.